First of all, the enzyme polymerase is an enzyme located on the DNA strand which forms particular polymers of either DNA or RNA. Today we will focus on DNA strands. The polymerase chain reaction is when these enzymes called polymerase take place in a reaction to produce many copies of a specific target segment of DNA. To perform the PCR, these following ingredients are required: a template DNA, of course, a 25 micrometer magnesium chloride, which is an essential cofactor for Taq polymerase. Taq DNA polymerase is an enzyme that helps catalyze the polymerization of the deoxynucleotides in a DNA strand. A pair of primers. A primer is a single-strand DNA sequence designed to locate the target DNA factor. 2 micrometer DNDP mix, which is a mix of nucleotides which are the building blocks of new DNA strands. And finally, the 10 times PCR buffer. It creates an environment for optimum activity of Taq polymerase. Now we will shift to the steps of the polymerase chain reaction. The target segment of the DNA is indicated in red. This part is the part that we want to copy. Primers and nucleotides in Taq polymerase have to be present. The first step of the polymerase chain reaction is denaturation. We need to denature the target DNA strands by applying heat. The heat applied is 95 degrees Celsius. This will break the hydrogen bonds. That's why Taq polymerase is important in this step, because it endures the scorching heat. The second step will start when the temperature is decreased to 60 degrees Celsius so that the primers can form hydrogen bonds. Primers attach or anneal to DNA primers required to attach to each strand of DNA. Primers are needed to be added so it can give the Taq polymerase a starting point. Note that primers follow base pairing rules. The third step of the polymerase chain reaction is elongation, or some people may call it extension. Taq polymerase binds to each PCR primer and begins adding nucleotides. Elongation will occur. This step requires a temperature of 72 degrees Celsius. After cycle 1 ends, 2 molecules are copied. But PCR mixture contains mainly copies of primers and a lot of nucleotides to perform additional cycles that will give even more copies. So when cycle 2 is performed, there is 4 molecules of DNA, and after 3 there is 8, and only 2 of the double-stranded copies contain the target segment. After many cycles, the number of copies increase exponentially. After cycle 25, as we can see, 33 and a half million copies of target DNA are produced. The exponential growth of the copies are very important because they are used in biochemical labs. Scientists model an equation for the growth of the copies, which is y equals 2 to the power of x, where x is the number of cycles and y is the number of molecules copied. In the graph, the x-axis represents the number of cycles and the y-axis represents the number of molecules. There are many applications of the polymerase chain reaction, but in this video we will explain the very important ones. First, analysis of DNA. Second, tissue typing. Third, PCR cloning. 4, production of hybridization probes. And 5, genetic fingerprinting. After PCR, the copied segment of the parental and offspring DNA or DNA from ancient sources are analyzed. This can be used to confirm the biological parents of a child or identify the remains of an identified body. It also detects viruses and diseases. This is an example of a blastomere cell being analyzed. Tissue typing is the assessment of the immunological compatibility of tissue from separate sources, particularly before organ transplantation. Histocompatibility antigens are molecules on the surface of all cells. Each person has a unique set of histocompatibility antigens. If the antigens of tissue or organs from a donor do not match the recipient's, a rejection response will occur. Generally, typing is performed on blood cells. Specific antibodies react with the cells. Each antibody preparation is specific for one histocompatibility antigen. If the antigen is present, the antibody will bind to it. Class II antigens are determined by the polymerase chain reaction. This is an example of the kidney transplantation. Despite PCR's speed and specificity, application cannot substitute for gene cloning in cells when large amounts of a gene are desired. Occasional errors during PCR replication impose limits on the number of good copies that can be made by this method. When PCR is used to provide the specific DNA fragment for cloning, the resulting clones are sequenced to select clones with error-free inserts. Gene cloning is a process that produces many copies of a gene of interest. These copies can be used in sequencing the gene, in producing its encoded protein, or in basic research or other applications. In this example, hummingbird genes are inserted into plasmids from E. coli of the plasmid, and the mixture of millions of different hummingbird DNA fragments would be present in the samples. These are the steps. DNA hybridization. At high temperature, double-stranded DNA will denature or separate into single strands. When the temperature is lowered, the two strands will anneal because of the base pairing interactions of the complementary strands. If the nucleotide sequences are similar, complementary strands from different sources will also anneal. This is called hybridization, to indicate that each strand of DNA came from a different source. Regions of double-stranded DNA in which the respective single strands hybridize to one another are said to be homologous, or have similar or identical nucleotide sequences. The high specificity of base pairing interactions between complementary strands of DNA can be used to locate a specific nucleotide sequence in a sample. If the DNA from one source is immobilized by attachment to a solid surface such as nitrocellulose, homologous DNA from another source will hybridize and be retained by the immobilized DNA. This is the basis for various probe techniques. Non-homologous DNA will not attach. This produces hybridization probes for both northern and southern blot. Southern blot is a method used in molecular biology for detection of a specific DNA sequence in DNA samples. Northern blot is the adaptation of the southern blot procedure used to detect specific sequences of RNA by hybridization with complementary DNA. DNA fingerprinting is basically examining DNA by using PCR and gel electrophoresis. PCR amplifies the fragment targeted, and then in gel electrophoresis, agarose gel and electricity separate DNA fragments based on size. Researchers and scientists later analyze the DNA. Thank you for watching my PCR video.